Wow, that was an excellent talk. Really excellent talk. I didn't know I was going to have to follow such a great talk. I appreciate that talk. My name's Hunter Land. Thank you for having me again this year. Thank you to the organizers and all the beautiful people here. I'm going to speak today about some advancements in cannabinoid-based research and give some insights from rigorously designed studies and also some not so much rigorously designed studies. Let's see. That was quicker than what's happening. Okay. Now we go. All right, a couple disclosures. I've been in the industry about 20 years, 12 focused on cannabinoids since starting with GW Pharma in 2012. I'm the VP for BRC. I also act as the Chief Scientific Officer for the NHL alumni, as well as do some lecturing and sit on some other boards. But now, on to the interesting section. So first I'm going to talk, for those of you that don't know, the FDA recently put out a publication on an update on INDs around cannabis and cannabinoids. So I think it's important to just run through that to give you kind of a glimmer into where the FDA is with current cannabis thinking. Then I'm going to talk about the challenges of research. I think many of you are aware of these challenges, but I'm going to put some of them, not all of them, just kind of in one place to help us understand how we really should be evaluating these publications and how we may use that information and how that information may bring more questions. And then finally, I'm going to dive into kind of the clinical trial update, talk a little bit about the landscape, and then finally, I'm going to discuss the trial that we have ongoing, one of the four that I can speak about today. I had hoped to provide you with data on this study, but as you know, contracting with lawyers can take a little bit longer than one might expect. So. All right, so the FDA, long history with FDA and cannabis. We've got about 800 INDs that have been, keywords, submitted since 1970. 400 from 1970 to 2010, and then another 400, so doubles from 2010 to 2023. This represents about a 300% increase in the last 10 years. And they did state, kind of in this small area, many submissions were unable to meet the IND requirements. So they didn't really tell us how many were unable to meet, but many. And one thing I'll just point out here is they have this nice graph, and you can see this section over here, addiction and pain, is kind of lumped into one. So I have some questions about, how much of this is abuse liability work versus actually therapeutic treatments. I'll dive into that in just a minute. Then they had this other very interesting, lovely graphical presentation of the changes throughout history. So we have a lot, the first 2 decades all smoked, and then we start seeing some other formats come in, and then lo and behold, look at this, like almost everything you can think of. We've got sweets and baked goods. So a lot of INDs going in. I don't know how many of these have been approved. I know some of them have, but a lot of INDs going on, different formats, different constituents. So again, interesting. So I have questions, which is normal for me. Anytime I read anything, I always have questions. Some of the questions I have here is how many INDs around cannabinoids have actually been approved? How many of these studies are looking at therapeutic benefit versus risk? I mean, I see a lot of studies on driving or, do you prefer this drug over that drug? Is it addictive, et cetera, et cetera. So again, if we're looking at cannabinoid research for therapy, it may be smaller than represented here in this particular publication. And then secondly, I would say, how many of these IMPs or these investigational medical products could actually become a pharmaceutical? So I know from conversations with the FDA, smoked cannabis or even vaporized cannabis, the VOCs emitted will most likely not ever meet the level of an approved drug. So while it's great, And there is, a very nice pathway to using medical cannabis as an inhaled product as far as being able to get that approved by the FDA and reimbursed by insurance. It does not appear currently likely, and we can argue about the merits of that if you want, but that's the current FDA standing. So maybe an inhalation device, but again, I'm not aware of that technology. Certainly, I think it would be tough to have FDA-approved THC cookies and stuff like that. So a lot of questions. And then if you dive down a little further, then you wonder how many of these INDs were for specific products. So typically, we would attach an investigational new drug application to an indication. So for Epidiolex, there's Dravet, there's Lennox-Gastaut, there's tuberous sclerosis, and a number of other indications like Parkinson's and several other research studies that have used Epidiolex, which would have different IND numbers. So we could have at least 10 or 15 different INDs for Epidiolex, same thing for Marinol, same thing for Sativex. So the bucket of actual IND research for therapeutics may be much smaller than is exhibited in this particular paper. But what this does tell me is that the FDA is supportive of cannabinoid research. We clearly have INDs, whether it's for therapeutic benefit or whether it's for abuse liability, that have been approved. I personally have these approved right now and have not had a big difficulty with the agency, although you do have to respond very quickly to their questions, and that may be cumbersome at times because they give you a couple days, maybe, to give you those responses. You must meet the CMC standards. Most people can't meet those standards, but it can be done, and you have to have sufficient documents. So investigator brochures, protocols, ICFs, and if they don't like something, like recently they told me blood pressure above 150 needed to be blood pressure above or below 140. So they do make these kind of suggestions around protocol design, not just CMC, et cetera. And then there are just some forms, 1571, 1572, 3674, et cetera. So it is a process. There's a 30-day review period. As long as you don't go on clinical hold after 30 days, you're free to run the trial. They could put you on clinical hold at any time, but we haven't seen that. So just make sure you have sufficient time to respond to these questions. All right, so now we've got that out of the way. I'm going to talk about challenges. I spend several hours a day. I have alerts on Google with every new study that gets published. I work with researchers. They tell me, what they're working on and when it's going to be published. So I look at these and I try to glean knowledge about how this could translate to research in certain indications. And the main thing that I find that just is added variables for me understanding how to use these data are things like route of administration, the product consistency, the format. Is there a control group, and how does this impact the interpretation of data? So I say this often, cannabis is not cannabis is not cannabis. I think we all know that here. But when you see a paper that says cannabis treats pain, I'm like, okay, what cannabis are you using and what pain are you studying? So there are a lot of different variables just in that example. And you get significant variability based on route of administration. So smoking for pain is different than oral administration. Certainly vehicles can have a big difference. So if you look here, this is SEDS versus MCT. For the record, MCT bypasses lymphatic absorption and has the lowest bioavailability of any lipid that you use. Whereas you can see rapid absorption with SEDS. Not saying you should use SEDS, but clearly there's a difference. So if I take 5 milligrams of THC and it's in SEDS format versus MCT, then I can expect a very different effect. And that may lead to efficacy, right? So 10 milligrams is not 10 milligrams is not 10 milligrams. And then finally, lack of proper controls. And that's kind of the outlier here, but what I want to say is we see a lot of studies that are where we look at research and it's with no control. So kind of proof of concept, does this work, right? So let's say 10 patients. And that's great because likely at a minimum, you're going to get a placebo response, 20%, 30%, 10%, something. People just Even in studies where people say, we're going to give you a placebo and rate your pain, people still get reductions in pain, even though they know they're taking a placebo. So in this case, we're giving them cannabis, so there's even a higher potential for a placebo effect. The problem is, yes, you may get a win in your study, but if there's a side effect, let's say you get a skin rash and there's 10 people, well, then all of a sudden, that's disproportional to what may actually happen. So if you have a control group and you get 4 skin rashes, well, maybe it's just the clinic and there was a skin fungus going around, right? But if you don't have a control, then essentially all that could be contributed to your drug. And as you know, if you use these types of studies to support a sponsor IND, then the FDA very much could say, well, you know, there's some serious safety concerns here based on your adverse event profile, because you have no control to say, well, actually, this is the same as placebo. That is a laser. Sorry for Bonnie, I think I just shot you with the laser. Okay, so this is Sativex. So I try to pull at least one variable out, right? So this is a known medicine, it's consistent, it's Sativex, and the title was effective cannabinoids on neuropathic pain. And so I dove in, and you can see here pretty clearly you get some separation. So here's the line, and then as you move this way, favors intervention. So looks pretty good generally speaking. I dove in a little bit deeper, and even though they're under the guise of neuropathic pain, they're actually quite different indications. So chemo-induced peripheral neuropathy, rheumatoid arthritis, like clearly different pathophysiologies there, MS-related pain, allodynia, diabetic peripheral neuropathic pain, MS central neuropathic pain, and peripheral neuropathic pain. So the same but different, and we're seeing different changes. So again, if you look here, DPNP, not so good, but if I looked at MS pain and I just assumed that Sativex would work for this, maybe not as much, right? Maybe we have to be a little bit specific and think about the etiology of pain. And maybe it does work. Maybe it was a study design issue, but I don't think we can just say broadly, this works the same across these conditions. Clearly, there's some variation. All right, this is a really interesting study, kind of builds on the indication. This is Cersei's work, and I haven't seen Cersei here, but I love this study because I keep seeing work on CBD, and a lot of times the doses are what I would say are far subtherapeutic. So what she did is she looked at all the RCTs, and the green dots are success. You can see the indications here, and the orange dots are not successful. And what she found is that the doses typically for success acutely are between 300 and 1,200 milligrams a day. And when you have repeat dosing, that goes down a little bit. So you can see repeat dosing, you probably get kind of a higher level of steady state. So this more or less confirms for most conditions, you need much more CBD on board, or at least I would say to optimize CBD's effect, I think we need much more CBD. And you can see sometimes you use these high amounts and they still don't work, right? And I think that like schizophrenia, I think they're missing a couple positive studies there. Again, it's not my paper, but you can see the general trend, and so it makes me a little bit frustrated when you see a study that, you know, we've used the subtherapeutic dose. I mean, the worst thing in research for me is when you don't answer any questions. A failed study, at least you know, when it's convoluted and you have no answers, that's a problem. And then if you look at this bottom corner, when they correlate to plasma levels, again, not many studies, but you can see plasma level versus success looks much better here than the low-dose studies. So again, maybe underdosing CBD. All right, this is one of my favorite sections to talk about, and nobody really likes statistics, or statistics, as many people say. So in research, I see just continually, especially in cannabis, I see these things. It's like statistically improved something. And you look at the p-value and you're like, oh my gosh, p-value less than 0.5, this must be real. And it is real, right? Like, no doubt, I mean, you can question some of the data, but if you're getting positive p-values, that's a positive thing. So basically, as most of us know, this indicates whether the observed effect was just by chance. And we test it with ANOVAs or t-test, right? Typically, the threshold's about 0.05. For those of you who don't know, that basically means if we ran this study 100 times, then we would expect this effect 95 times, which is pretty good. The odds are in your favor that this is a real effect. But it doesn't convey practical significance or the magnitude of effect, which I think is something we often miss. On the contrary, effect size means how big is the observed effect, which is great, which could be statistically significant or not based on the data. And it gives real-world insight. So like Dustin, if he sees a paper and he's seeing a 50% improvement in something and statistical significance, he's probably going to feel a lot better about that paper than if he sees a 5% improvement with statistical significance. So I'd say clinical meaning of seeing this magnitude, very important for clinical practice, certainly important for research. So as I mentioned, you can have something that works that doesn't have a big magnitude. You can have something that has a big magnitude that's not statistically significant, so we still don't know if it's by chance or not. And that's one way they work together to interpret studies. Now, next I'm going to talk about a study that I was excited about. So if you guys know me, I love minor cannabinoids. I love the idea of rational polypharmacy or super additive effects or potential entourage or non-entourage. So when this came out, I was like, oh, great, what happens? CBN and CBD or CBN alone, what happens in sleep? So I looked at it. It's a double-blind, randomized, placebo-controlled study. They looked at a 20-milligram gummy of CBN. I thought that was low. Most of the binding says it's like 1/20th the potency of THC, but that's aside. But I see these numbers, and this is kind of verbatim from the paper. It did not hit on primary endpoint, but they said significant improvements in reducing awakenings, 0.05, significant improvements in sleep disturbance, 0.02. Okay, I'm excited. Let me read more. So I go and I look, and they have these graphs. I look here. This is the CBN group alone, and these are the combinations. And you can see this has moved over. I look at the scale, difference from placebo. Let's see, right? And I come up here, and it looks like it's about a half, but clearly over this line, right? So I look down at the next one, and I come over here, and I'm like, okay, good, about another 1 difference from placebo, the scale from 4 and 2, okay? So I'm like, okay, well, that's interesting. Not familiar with these scales. Let me look into the scales a bit. And ultimately, what you see is the first scale was awakenings, right? So number of awakenings. So we had a reduction of 0.5 awakenings a night. So every other night you wake up 1 less time. Maybe that's meaningful, maybe it's not. It's not a lot of awakenings, but to be honest. And then I looked at the scale they used for sleep disturbance, and their baseline was 15.1, and I think we landed at about 14. So this is a 20-point scale. So we got a 1-point reduction in a 20-point scale. Statistically significant, but like I don't think that we can say that 20-milligram gummy is now like the best new sleep aid. Like it's not that much. I suspect if you would have done this with delta-9 THC, we would have a much better, much more interesting result than this. Maybe if we went higher with CBN, we would have a much better result. Don't know. But so what do we have in this study? We have a control group. That's great. We've got a control group. I'm happy with that. Format, I don't know if 20 milligrams of CBN has gotten into these patients or not. Like I have no idea, because I know if you put it in syrup and you eat it, I mean, Ryan Vandry did that study with CBD and jam, and like it was non-detect in the blood. So I don't know, is this a SEDS gummy or is this just like a gummy? Indication, it was self-reported poor sleep. So do you have sleep apnea, restless leg syndrome, or are you just having trouble sleep? So again, I don't know this piece either. And then finally, does it have both statistical significance and a meaningful effect size? And I just don't think so. I wanted something really new and exciting with a minor cannabinoid, and CBN is available, right? It's a degradation product of THC. It's just old THC. So it's easy. We could easily research this. It's not like we're looking at CBT, which is really hard to get. It's like, oh, we could get this. But based on this, it doesn't look exceedingly positive. All right, so let's talk about some rigorously designed studies. Not all of them are positive. This is the first one that was related to kidney stone pain, and it was a 20 milligram CBD intervention, which just makes me sad because they had funding for 90 patients to do a 90-patient randomized controlled study, and they used 20 milligrams of CBD for pain. Everything was standard. It was designed well, just the dosing wasn't right, and basically we saw nothing. In fact, placebo looked slightly better in all these outcomes, and it makes me sad because, we could have used something else or something more than 20 milligrams CBD. But well-designed study just didn't get a cannabinoid person in there to say, hey, we should talk about dosing. Another one. This was a alcohol use disorder study with PTSD. They used a dose that I would think would be in the range to see efficacy if it works. 600 milligrams CBD, pretty robust study. They did not give the stats. So I got this from clinicaltrials.gov. It's not been published, but they gave the outcomes. And just based on looking at the outcomes and drink reduction and heavy drinking days and patients without heavy drinking, we're really not seeing anything. We see a little bit with the PCL-5 score at the end, which isn't related to drinking, that's related to PTSD. But generally speaking, no big hits. Based on this study, I wouldn't suggest that CBD is going to stop people with PTSD from drinking. It doesn't look particularly positive. So again, well-designed studies. I think we answered the question, or at least to some degree answered the question. Had there been THC present or CBG present, this may be a very different outcome. But CBD alone, not looking good for alcohol use disorder. Also, we've got recently terminated studies that were well-designed. So the first one was the release study, the MS study, spasticity. They were counting spasms, which actually counting spasms is quite hard because it's not like seizures or most seizures. It's difficult. And the primary analysis did not show positive, was not positive in counting spasms. So Jazz pulled the study. Next, we went to this myoclonic atonic seizure study, which looked to get, there was a big focus on cognition improvement and behavior in the study, so I was excited, and that was also just pulled. So the two big Jazz studies that were robustly designed as phase 3 will not have data. And then there's the Prader-Willi syndrome study, RADIUS, and this was pulled due to change in corporate priorities. So this study was voluntary terminated. So again, another study we could have looked forward to, answer some questions, not there. So all kind of sad and disappointed. But I think that there are other studies coming. I saw new studies listed, so I think there is an influx coming. This is kind of the trough, and there's a delay, and there's some more stuff coming. One that I wanted to talk about, which is interesting, just kind of came out of left field, that is positive. was a study on dental pain, acute dental pain. I've never done anything in dentistry. I never thought to look in dental pain. And they looked at Epidiolex, 10 mg per kg and 20 mg per kg versus placebo, 20 patients in each group, enough to see signal. I don't know, there was like the somewhat passive-aggressive comment at the front. It said, GW had no participation in the design or conduction of this trial, nor did it provide any funding or material support. So that leads me to believe they might have submitted a request that got rejected and just did it on their own. But anyways, typical endpoints, pain relief, bite force, and then looking at like psychosis or psychoactive effects and mood changes. And what do we see? Pretty cool. So this is the non-normalized to placebo, but you can see a pretty significant drop in dental pain between these two groups. This is placebo, 10 and 20 mg. When they normalize, you can see it's pretty clear. And I would say not only is this statistically significant, but there is a nice magnitude of effect here. Like this isn't just like a 10% reduction. It's a pretty big reduction, not from baseline, but from placebo. So I'm pretty happy with these results. They also looked at bite force, which is much more of an objective outcome. And it's tough to see here in this graph because you do get variability. But when you go over here, you clearly see that this 20 mg per kg is separating from here in terms of bite force. So not only do you have self-reported pain, but you actually have the mechanical act of saying, hey, does this hurt when you bite? And you can go to a much higher level because it's not painful. So interesting. Maybe dentistry, maybe there's needs to be more work in the dentistry segment. So also need to talk about some of the adverse events. We didn't really see any psychoactive changes. By that they mean intoxicating, no mood changes. Saw some sedation, so we've seen that before. Actually more in the 10 mg than the 20. Don't know what to make of that. Diarrhea, that's been reported. Same thing with abdominal pain. So just to be clear, it's not the oil that gives the diarrhea. It's something going on with CBD. Most of this is transient, and at least in epilepsy, a lot of the patients were like, well, these AEDs make me constipated, so it's great that I'm getting more regular. So side effect could be benefit in some cases. All right. Last study I'm going to mention is an older study. This is Zynerba's study. This was a topical CBD study. It's a little bit older, and you may be wondering why I'm going to present this study. First, I think that exposure, they never provided their PK for this study, so I think exposure to CBD is fairly low unless they took a shower in the Zynerba topical. And in the first group, you didn't really see much combined, no p-values. But if you look at this 90% methylated group, you do start seeing positive p-values. This is social avoidance. So for those that you that don't know Fragile X, it's like kind of like a developmental encephalopathy. They have often have seizures, but also like autistic-like behavior. So this is a win, but it only happened in this group. And again, older study, and this is kind of Brian Thomas's brainchild. So if you know Brian Thomas, he's a legend in the field, and usually have to get a whiteboard out when we have conversations because it's a lot to digest, and things make connections that you don't expect to make connections. And as we move on, again, you see in this group, you can see these trends, and again, statistical significance and social avoidance. And I think this is a very low exposure CBD. I would have loved to have seen what happened with 10 or 20 mg per kg CBD, or with a combination product that had a little bit of THC in it. But again, looking positive. So why is this? Why are we seeing this fully methylated group? And we could say maybe they're affected to a greater degree than the other group. Maybe that's one answer. But the other thing it could be is FMR1 leads to the loss of FMRP and overexpression of DAG lipase and 2-AG. And when that happens, so some people may say that's good, but actually what happens is you tend to desensitize CB1, right? And when that happens, it's a loss of function. So by increasing these endocannabinoids constantly, you've desensitized these CB1 receptors. By adding CBD, Robert LaPrairie and Kim Mackey have done a lot of work on negative allosteric modulation. It may be less is more, because what seems to be happening is that seems to interfere with beta-arrestin-induced internalization. So while you may decrease the binding of THC or the endocannabinoids, it may actually increase overall CB1 expression, leading to more receptor sites. So less may be more. We know that the strong agonists like spice and K2, not good drugs, actually quite toxic. So we may actually want a weaker binding agonist, and we may want to retain this CB1 tone. And so I bring this up because this may be, as Ethan has spoke about, and we heard with endometriosis, like we actually may want a weaker agonist than a stronger agonist to target this area. And I am almost done, so I will briefly talk about, we've got study just started. I think it's an innovative study that's clearly biased because it's my study, but it is mechanisms with high CBD extract. This is a botanical drug substance. It's in complex regional pain syndrome. And as you can see, complex regional pain syndrome looks terrible. I don't have to tell you all the details, but usually it happens after a fracture. Nothing touches this condition other than maybe nerve blocks. It's got autoimmune component, bone component. It's got a lot of problems, not a lot of things to do. And they're really sensitive to temperature. So what we're doing, which I never really got to do before, is we're looking at pain, but we're looking at pain under fMRI as well. So BOLD looks at the oxygen level. So if you see healthy individuals versus chronic pain, you see a significant difference, so a very objective outcome. And we have placebo groups, so we'll see what the changes look like with and without medication. We're also doing the standard treatment for 8 weeks, and we're looking at things like the standard VAS scales, a brief pain index. We've got depression, anxiety trait, sleep, PROMIS 29, some trough pharmacokinetics, and then a bunch of biomarkers related to pain and inflammation. So very excited, hope to present the data for these today, but I don't have it yet. So, and we've got 4 more of these coming where we've got approved INDs. So that's what I'll leave us with today, and thanks so much for your time. I don't have time for questions, but if you guys like to chat afterwards, please let me know.